Okay, so today we're going to talk about the relationship between amino acid and citric acid cycle and metabolism in general. So let's think back to what we know about amino acids, which we spent a lot of time, so hopefully you won't forget that part for your future classes. So we always hear that amino acids are the building blocks. And so the carbon skeletons of the amino acids, they will go and be reshuffled for other molecules. So they will look at tissue proteins, enzymes, structural proteins. Okay, they can also go into forming other molecules, and the ones that we're going to focus on are the ones in the citric acid cycle. So before you panic on this slide, I won't ask you specifically which amino acids go where. It's more understanding the overall concept of it. So the carbon part can get reshuffled into different intermediates. So for example, glutamine, arginine, proline, histidine can form alpha-ketoglutarate, one of the citric acid cycle intermediates. Tyrosine, phenylalanine can form fumarate. So I won't go through all of them, but you see how it's linked up. So catabolic breakdown means that the amino acids break down into citric acid cycle intermediates. And then the other way is true too, that some of the citric acid cycle intermediates can form some of the amino acids. And here's what's really interesting is I'm going to make kind of a big deal about that part here in a second, is the idea that you see this arrow here. Some amino acids will form pyruvate, and then some amino acids will form oxaloacetate. There's no arrow there. But what's interesting about that will be how that plays into metabolism when we have low blood sugar, low glucose, and we need energy. Okay, so here's that link that some amino acids are said to be glucogenic, which means that this carbon skeleton forms pyruvate or oxaloacetate. And both of those guys, if you recall, can go into gluconeogenesis. And then some amino acids can form acetyl-CoA or acetyl-acetyl-CoA, and those will form ketone bodies. So amino acids can be glucogenic, ketogenic, and some actually can be both. Okay, we'll come back to that top sentence here in a moment, which is about transamination. We'll come back to that one, so hold on to that thought for a second. So here's a listing of our amino acids. Most of them are ketogenic, meaning that they can enter in as pyruvate or oxaloacetate into gluconeogenesis to form glucose. And a couple are ketogenic only, and those can form acetyl-CoA and then form ketone bodies. And then you have ones that can do both. Okay, and this is a really important slide, not for you to memorize which ones are which, but to understand that when there is low amounts of glucose in the body, because you haven't eaten for 4 to 6 hours, you're entering into that mode where gluconeogenesis is going to kick in, then you can start pulling from your proteins to make glucose or your proteins to make ketone bodies. So this is something we will come back to in our next in the next power video lecture. Okay, so going back to that other comment that Here's our amino acids. I just said that this carbon skeleton was going to go and either form tissue proteins, structural proteins, enzymes, citric acid cycle intermediates. The other thing that could happen here is that when that happens, the NH3 group has to go somewhere, but it's toxic. So ammonia is toxic. It cannot travel through the bloodstream on its own. So there's this idea then that the ammonia will get transferred to other molecules. Alpha ketoglutarate is one of the main ones, and that process is called transamination. It's the transfer of ammonia onto other molecules. When it gets transferred onto alpha ketoglutarate, then it can go through the bloodstream. Usually that transformation, alpha ketoglutarate, goes to glutamate, and you don't need to know that specifically. But once it gets transferred and you form a new molecule, that molecule now is a safe way for the NH3 to get transferred through the blood. And then ultimately, it could be used to make other molecules and other pathways, or it can be excreted out, and that gets shuffled through urea cycle. And ultimately, we would excrete that out. And that urea cycle, I'm just going to show you a pic. You don't need to know it, but it's mostly to understand that NH3 group, what has to get transferred on. Alpha ketoglutarate, our main molecule that does that, best example of it. And then that now can flow through the bloodstream. Ultimately, it could be used for other molecule synthesis, but another thing that will happen with that NH3 group is that ultimately it's going to be converted to urea, and then that will get excreted out. And that all of what I'm describing is the complexity part. I'm just looking at this in concept, so keep that in mind. This is just a concepts thing. All the nitty gritty is in chapter 23. If you maybe just kind of look at it, just kind of flip your book through it, and you'll see that it's very complicated. And kind of going back to our previous slide, why would it be complicated? Is you see every single one of our amino acids. Well, it is going to have its own pathway, its own set of enzymes that's going to get it to forming pyruvate, oxaloacetate, acetyl-CoA, right? That doesn't happen just easily. Each of our amino acids has its own path to get to where it's going as far as either being glucogenic or ketogenic. And so as you leaf through your book, you'll see every single one of them has its own little pathway. And so then it adds enormous complexity onto that chapter. Okay. So here's urea cycle. You don't need to know this specifically, but I just want you, the thing that I really want you to know is that proteins then, protein metabolism is linked to the excretion of urea. And this means that there's a whole process here related physiologically to the kidneys. So the greater amount of protein that you have in your body and the more that it's being broken down, then you're going to see an increase in your kidneys' function to get rid of that excess ammonia. And that would be the fine print of the diets that are so heavy on proteins is that they can, if people don't do them correctly, put a strain on the kidney because you need to drink a lot of water with that kind of diet so that you don't dehydrate. And this just has a little bit more detail regarding um, the urea cycle and how it's linked to citric acid. But I'm not going to ask you for this type of detail. But I just want you to appreciate that it's additional pathways and has a complexity to it.